O negative is often called the universal donor blood type, but its real story begins tens of thousands of years ago. Blood type O is considered the oldest of the ABO groups. Genetic research suggests it was the dominant blood type in early human hunter-gatherer populations. Unlike A or B blood, type O red blood cells do not carry A or B surface antigens, meaning they lack the specific sugar molecules that define the other groups. O negative goes one step further. It also lacks the Rh factor, a protein discovered in 1940 by Karl Landsteiner and Alexander Wiener. Without AB or Rh markers, O negative blood can be transfused to nearly any patient in emergency situations, which is why hospitals depend heavily on it. From an evolutionary perspective, some studies suggest type O may have offered resistance advantages against severe malaria. Research from institutions including Harvard School of Public Health and studies published in Nature Medicine have shown lower risk of life-threatening malaria complications in type O individuals. However, type O has also been associated with slightly higher risk of certain ulcers, particularly those linked to H. pylori bacteria, according to long-term epidemiological research. Genetically, O negative is inherited when both parents pass down O alleles and the Rh negative gene. It is recessive, making it relatively rare globally. O negative is not a superpower. It is a biological variation shaped by survival pressures over thousands of years, and today, it remains one of the most medically valuable blood types in the world. A positive is one of the most common blood types in the world. Its story begins thousands of years after type O first appeared. As human societies shifted from hunting and gathering to agriculture, genetic variations began to spread. Blood type A emerged from a mutation that added a specific antigen, known as the A antigen, to the surface of red blood cells. This antigen is a small sugar molecule that changes how the immune system recognizes cells. Being A positive means two things. The red blood cells carry the A antigen, and they also carry the Rh factor, a protein first identified in 1940 by Karl Landsteiner and Alexander Wiener. From an evolutionary perspective, some researchers suggest that type A may have become more common in densely populated farming communities. Certain studies, including research published in journals like Blood and epidemiological data from institutions such as Stanford University, have explored links between type A and immune responses here to specific infections. For example, some data suggest individuals with type A blood may have slightly higher susceptibility to severe forms of certain viral infections, including early COVID-19 studies published in the New England Journal of Medicine. At the same time, other studies indicate different inflammatory response patterns that may have offered advantages in past environments. Genetically, a positive is inherited when at least one parent passes down the A allele and at least one parent contributes the Rh positive gene. The A gene is dominant, meaning only one copy is required for the trait to appear. A positive is not better or worse. It is a variation shaped by human migration, diet, and population density over thousands of years. A negative blood type carries the A antigen, but it does not carry the Rh factor. This small difference makes a negative much less common than a positive. The absence of the Rh protein is due to a genetic variation in the Rhd gene, which prevents the Rh antigen from forming on the surface of red blood cells. Historically, the A blood group is believed to have emerged after type O, likely during the rise of agricultural societies. As human populations became denser, exposure to new pathogens increased. Some evolutionary biologists suggest that certain immune response differences linked to type A may have played a role in survival patterns during that period. However, the Rh negative trait appears to have evolved separately and is more common in European populations. Research in population genetics, including studies published in Human Genetics and data analyzed by institutions such as the University of Chicago, show that Rh negative frequencies vary significantly across regions, suggesting migration and genetic drift influenced its spread. From a health perspective, studies have explored statistical associations between A blood type and certain cardiovascular or inflammatory risks. For example, research published in Circulation has examined links between non-O blood types and clotting factors. But these are population-level correlations, not predictions for individuals. Genetically, a negative occurs when a person inherits at least one A allele and two copies of the Rh negative variant. Because Rh negative is recessive, both parents must carry it for a child to be Rh negative. In transfusions, a negative blood can be given to A or AB recipients who are Rh negative, making it medically important, though far less common than O negative. A negative is a subtle variation, shaped by migration, mutation, and immune system adaptation across thousands of years. B positive carries the B antigen on the surface of red blood cells, along with the Rh factor protein. The B blood group is believed to have emerged after type A, likely during periods of large-scale human migration into Asia. Genetic distribution studies show that blood type B is especially common in Central Asia and parts of India, suggesting evolutionary pressures in those regions influenced its frequency. The B antigen is a slightly different sugar molecule from the A antigen. This small molecular difference changes how the immune system identifies cells. Because of this, individuals with type B produce antibodies against type A blood, a principle discovered in 1901 by Karl Landsteiner, which laid the foundation for safe blood transfusion. From an evolutionary perspective, researchers have explored possible links between type B and resistance patterns to certain infectious diseases. Some population-level studies, including research published in Infection, Genetics, and Evolution, have examined correlations between blood group distribution and regional disease exposure. There is also ongoing research investigating how ABO types influence inflammatory responses and clotting mechanisms. Being Rh positive means the red blood cells also carry the Rh protein. The Rh factor plays a crucial role in pregnancy compatibility. If an Rh negative mother carries an Rh positive child, immune complications can occur, a condition now preventable thanks to medical advances developed in the mid-20th century. Genetically, B positive occurs when a person inherits at least one B allele and at least one Rh positive allele. Both the B gene and the Rh positive gene are dominant. B positive is not rare globally, but its regional concentration tells a story of migration, adaptation, and immune system diversity shaped by geography.